Good afternoon, everyone. Welcome. Hi, look, I'm Pankaj Sar, Director of the Queensland Brain Institute, and welcome to the 10th Peter Goodenough lecture, the 2018 lecture. And I'd like to start by acknowledging the Turrbal and Jagera people, the traditional owners of this land that the University of Queensland is situated on. I pay my respects to their elders past, present, and those to come. And a special welcome to Ian Fraser, our speaker for the 10th Goodenough lecture. It's a pleasure to welcome Peter Hoy, President and Vice Chancellor of the University, Jeff McCall, who's Executive Dean of Medicine, Neil Matheson, and Clive Jackson, who are trustees of the Peter Goodenough Estate, members of the QBI Advisory Board, members of the community, friends, and members of the community. Look, it's lovely to have so many people here once again. So Peter Goodenough was born in the UK in 1936 into a farming family, and he lived through the Second World War as a kid, really, and he kept moving around, and he migrated to Australia in 1964 under the Assisted Passage Migration Scheme as a 10-pound POM, and they were called the 10-pound POMs because that's how much it used to cost to migrate to Australia with your passage being assisted by the Commonwealth. So he landed in Sydney, and he headed up to Queensland, where he went on to develop a multi-million-dollar civil engineering empire. Now, he had a lot of interest in Papua New Guinea and built quite a large empire there. And in Bougainville, he employed more than 100 people who were working in building roads, in trucking, and in prefabricated housing. So it's quite a wide range of interests, but he made quite a lot of money there. And Peter Goodenough was diagnosed with motor neuron disease in 2001, and he suffered for 3 years and died in 2004. So his personal battle with the disease led to an international bequest and established the laboratory here, which is the Goodenough and Wantock Research Laboratory in March 2008. So his initial bequest was $3 million, which grew to $10 million. And out of that, we established the Goodenough and Wantock Laboratories. And as a bequest in collaboration with the estate trustees, we endowed several scholarships for people from Papua New Guinea, one in nursing, one, I think it was in agriculture, to move here, and they were for a couple of years. This lecture, the Peter Goodenough Memorial Lecture, was established to commemorate his far-sighted bequest. So the inaugural lecture was in 2008, and it was given by Gus Nossel. And this is the 10th lecture, and last year's lecture was given by John Parker from Sluder Medical. So it gives me great pleasure now to introduce today's speaker, Ian Fraser. So Ian was also from the UK, born in Glasgow, and he obtained his medical degree from Edinburgh University and trained as a clinical kidney physician in Edinburgh. He moved to Melbourne to work at the Walter and Eliza Hall in 1980, where he had an interest in viral immunology. And he was involved in the early link between viruses and HIV. So that's one of his early claims to fame. But he developed an interest in the papillomavirus, and he noticed that it seemed to have a role in forming precancerous cells. He moved to the University of Queensland in 1984, where he continued to work on HPV, and in collaboration with Jan Zhao, developed a vaccine for cervical cancer, Gardasil. So Ian's group grew from being in the university here to being quite a large immunology and cancer group, which eventually transformed into the Diamantina Institute. So Ian has published over 300 peer-reviewed papers and had the recipient of many, many awards, including the Queenslander and Australian of the Year in 2006. He received the Prime Minister Prize for Science and the Bolton Prize in 2008. He's a Fellow of the Australian Academy of Science, Royal Society, and the Royal Society of Edinburgh. So he's current President of the Australian Academy of Health and Medical Sciences and member of the Commonwealth Science Council. And most recently, he has been appointed Chair of the Federal Government's Medical Research Future Fund. So closer to home, Ian and his wife Carolyn are the co-chairs of UQ's philanthropic campaign, Not If When, the campaign to create change. And Ian has worked tirelessly for UQ, raising money in this exercise. They've forged partnerships between many generous donors in our community and have a vision towards a better world through change. I'm sure you're all looking forward to Ian's lecture, like myself. Ian, welcome.
Well, first of all, thank you very much for the invitation to come and talk with you today. It's always nice to get a chance to talk a little bit about science, but also a little bit about the role that philanthropy plays in facilitating science. I have worked in the field of cancer research now for maybe 30 years, and most of the work that I have done has been helped by the philanthropy of others. So this will be a sort of lecture, theory and practice of cancer research. Starting sort of with a fairly basic introduction to what I think is really important about public health in the future. We face a major challenge in the 21st century that we have a population which is steadily aging, and as we grow older, we become more prone to disease. When these cartoons were drawn on a temple in Komombo in Egypt some 5,000 years ago, this is a lady sitting on a birthing stool, and these are the instruments used for removing bladder stones from the bladder. Schistosomiasis being a common disease 5,000 years ago in Egypt. The life expectancy, the average life expectancy at that time was about 40 years. And it stayed at 40 years for the next 5,000 years. Despite all of the efforts of physicians to try and improve things, life expectancy did not start to increase until about 1840 across the planet. And while you might want to think that this was all due to a medical revolution, actually it was not. It was nor was it due to the American or the French revolutions, but actually to the Industrial Revolution, which gave us safe food and water for the first time. And a large part of the gain in life expectancy up to the beginning of the 20th century was due to safe food and water. After that came, of course, prevention of infectious diseases through vaccines and antibiotics, and life expectancy across the planet. The other increase is mostly through other research. That sort of paints a rather gloomy picture that the research doesn't actually give us any benefit, but of course the reality is it's not about how long we live, it's the quality of life we live. And the most of medical research has been to try and help us live longer, healthier lives. The trouble with that is that it comes at a cost, and we're spending an increasing amount of our budget on health these days. So that most countries in the world spend about 10% of gross domestic product on health. And these are some figures here. The US is the exception. It's now up to 18% of gross domestic product on health. And of course, that creates a big problem. The more important point is that what we spend on health is increasing much faster than the growth in the economy. And that's obviously not a sustainable situation. So that we obviously need to get the benefit from medical research to try and fix that up. So the Economist takes a view that better health outcomes are driven by productivity and cost effectiveness in the health services. And I spent quite a bit of time working on that with the McKeon Committee about 10 years ago. And we produced this diagram, which basically says that there are some really good things you can do to keep people healthy. And then there are some less good things. And then there are some things that actually make the situation worse. The good things are the things that I'm interested in, vaccination programs and public information campaigns. The less good things, of course, are when things go wrong with drug treatment or if you get complications in hospital that you wouldn't like to have, like catching an antibiotic-resistant bacterium. That's a great view of things from the point of view of an economist, but of course we don't live in an economy, we live in a society. And society thinks much more about the actual benefits than the cost-benefit analysis. Having said that, I think it's very important that we understand that we do have to think about investing in a long and healthy life, and that we have to think about how we might achieve that. So we spend about $2.1 billion a year on prescription medications, which are supposed to improve our health. They have been developed to help sort out mostly chronic disease. And that a large percentage of our population use these, and they're heavily subsidized by government. But we equally spend a significant amount of money on over-the-counter vitamins and medications, which are of no proven benefit, and the amount we spend on them is exactly equal to the amount we spend on prescription medications, at least according to the Australian Institute of Health and Welfare. If that money were made available for health and medical research, just think how much better off we would be in the future. So this is not a lecture telling you what to do, but it is a message that says that we haven't yet got in society our values right about health and medical research. That's to come in the future. But what I really want to talk about is one particular example of research, the cervical cancer vaccine development, and just talk a little bit about how it happened. But I would like to start off by a slide which basically points out how the business of research actually works. If we're looking at research for a clinical problem, it almost always starts with the clinical problem. And it starts with somebody making observations on that clinical problem, either in the public health arena or as a clinician. And I was a clinician working to some extent in public health. That, we hope, then leads to basic research to try and better understand how this problem comes about. We need to understand the biology and we need to understand the pathology. And then, if all works out well, then that basic research will lead to some translational research, which will turn that knowledge into some practical outcomes for patients. And if you're lucky and you can persuade people to do it, the challenge then is to take the outcomes of that translational research and make sure they're used. And this actually can be the biggest challenge, and we need public health research and health services research to make sure that we can actually use the research work that is turned out through the basic and translational research. And of course, it goes without saying that all of this is driven by money, and it's important to understand that without the funds to do this, we can't get round this circle. And while about 2/3 of all the medical research that is done in this country is paid for through one or other sources of government funding, about 1/3 of it comes from philanthropic donations. And it's the partnering of philanthropic donations with the government funding that enabled the cervical cancer vaccine. So we have to understand how we pay for research and the treatments. Obviously, one part of it is if we translate it and use it, then it's possible that we will save money by doing things better. But the other side of the story is that we have to prioritize proven interventions over ineffective medication. And that's actually one of the big challenges that government faces now, is understanding what works and what doesn't. And indeed, a large or a significant part of the Medical Research Future Fund, on whose advisory board I sit, will be targeted at making decisions in this area where we can prioritize proven interventions over ineffective medications. If we spent all that money on vitamins, on research, as I've said before, we would be much better off. Okay, that's the public health lecture over and done with. Let's think about cervical cancer for a bit. Cervical cancer, this is a personal history. It's my point of view on the topic rather than the world's point of view, but I hope that you'll get some messages out of it. So, first message, 20% of cancer is caused by infections. That comes as a surprise to people, but it's the reality across the globe. 5% of that is papillomavirus infections. That's 1 in 20 cancers worldwide caused by infection with one single virus. And we recognize that this includes cervical cancer, but also cancer in other parts of the anogenital tract and also in the throat, oropharyngeal cancers. It's worth looking at the rest of the list. The ones that are in green on the list here are viruses where we actually have a vaccine to prevent them now. And we can therefore prevent about 8% of the cancer burden through immunisation. We have need of the vaccines that are listed in yellow there, although hepatitis C is getting a little easier because there are now drug therapies which are effective against chronic hepatitis C infection. But it would be better to prevent it than cure it. And then there are some interesting new viruses down at the bottom here. For example, Merkel polyvovirus has only recently been recognized as causing cancer, a rather rare cancer called Merkel's disease, which is a skin cancer. But there may be still further viruses to find. And then we have to recognize Barry Marshall and Robin Warren's work on Helicobacter, showing that this bug causes gastric ulcers. And then on top of that, we now recognize that globally it causes gastric cancer, and it's responsible for about 5% of cancer worldwide. So this is, in principle, all something we could do something about, because we know how to control most infections. It's worth pointing out that in most cases, the infections are common, and cancer is a rare consequence. So for papillomaviruses, I'll show you. So we all get papillomavirus infection, but only 1% of the people that get papillomavirus infection are at risk of cancer. And that is true to a large extent for the other viruses on the list there as well. Now, HPV-related cancer deaths are predominantly, but not entirely, a developing world problem. This map basically shows in red the countries where most of the cervical cancer deaths occur. And Australia is pink because we have a good screening program for cervical cancer, as is Canada and most of Europe. But sub-Saharan Africa, South America, India, China have major burdens of cervical cancer deaths. And 250,000 women die of cervical cancer every year globally. Now, if there was an epidemic of any other infectious diseases that killed 250,000 worldwide women or people every year, then there would be an outcry to get something done about it. But the reason why there isn't so much of an outcry about cervical cancer is because there's a long disconnect between the time when you get the infection and the time when you get the cancer. And the vast majority of people who get the infection never know they've had it. So that there isn't the great outcry until you start looking at a map like this and realize that 250,000 preventable deaths are occurring globally every year. So I said this was a personal history. We're going back a long way in time. We're going back to the beginning of recognition of cervical cancer. Back in the 1840s, it was the first time that people could even start to talk seriously about cancer. The combination of microscopy that allowed you to look at cells in the body and the dyes that the German dye industry developed for dyeing cloth, which allowed you to stain the tissues of the body so you could actually see down the microscope what was going on, enabled people to recognize there was something wrong with your cells when you got cancer. Prior to that, cancer had been recognized as a problem, but the cause of it was not clear. This guy, Domenico Rigoni Stern, was a mathematician in Verona, and he tried to work out who would be who got cancer, basically. And in those days, remember, the average life expectancy was still about 50 years, and there were only two common cancers. One was breast cancer, and the other was cervix cancer. Both occurred in women. So he did some studies on these, and he made the observation that cancers were more frequent in nuns than in other women, at a ratio of about 5:1. Now, you could only really do this study in Italy, because in Italy, 20% of the female population at that time were nuns. It's true. I mean, if you had a daughter, that was fine, you married her off. If you had a second daughter, then she might well end up in a nunnery. The frequency of cancers of the uterus, which is actually cervical cancer, was inversely related to that of cancer of the breast. Now, those stories are both absolutely true, and he produced the figures to show it. You don't need to read these. It just basically says that he looked at single women, including nuns, and married and widowed women, and basically worked out who got which cancers. Now, if he had stopped at that point, that would have been fine. He was a mathematician, and probably he should have realized that that was his limit. But he decided he would also be an epidemiologist and try and work out what that actually meant. So he decided the incidence of uterine cancer was not increased through the licentious practices of women. He was wrong. I'll come back to that in a moment. But the risk was greater amongst women who were excessively sensitive morally and nervously irritable. He didn't do any better on breast cancer. He decided that it might be due to the fact that the nuns' habits were too tight, or the long-sustained bent position where saying prayers, pressing on the breast, caused the disease. Now, these two statements are interesting, because if we're thinking as a scientist, that is at least in principle a testable hypothesis. The previous one about nervous irritability, I regret, is not. So we just have to accept that he didn't quite get that bit right. But he did have the right idea, and he was starting the business of epidemiology of cancer. The first real step forward with cervical cancer and controlling it was when this gentleman, Papanicolaou, came up with a way of screening for cervical cancer, the so-called pap smear, which at least the women in the audience will be familiar with. And what he did at that time was to develop this as a test for cervical cancer, not to prevent it, but to find out if it was there so it could be treated early. And he was looking for cancer cells looking like this in the pap smear, rather than these relatively normal cells from the neck of the womb, this one's actually infected with the papillomavirus, which is why that picture's there. But these are pre-cancer cells, and that's what the test was designed to do. He had no idea what caused cervical cancer. He just wanted to find a test to screen for it. At about the same time, the observation that Rigoni Stern made, that this was not a disease of licentious woman, as he put it, was beginning to be doubted in the sense that it was clear there was a connection between the number of sexual partners that you had and your risk of getting cervical cancer. And a number of epidemiological studies, including this one, were published basically establishing that there was a significant risk amongst patients who had cervical cancer having multiple partners in comparison with the controls who did not. So that was a step in the right direction, I guess. And the question then was, why was that true? An interesting sub-study that came out of that same study. Epidemiologists like to crunch numbers, so what they did was they noted that a considerable excess of patients who had their first sexual intercourse on the ground in comparison with in a hotel room were likely to end up with cervical cancer. I'm not sure that specifically helped working out why they got cervical cancer, but it was nevertheless statistically true. Association does not prove causation, very important in all of science. Just because you see something happening together doesn't mean the two are true. And that turned out particularly to be true when they started looking for the real cause of cervical cancer, because they noticed that there was an observation that this virus, herpes simplex virus, infections with that virus were much commoner in young women with cervical cancer than they were in controls. And for quite a while it was thought that that virus might well be responsible for cervical cancer. But it turned out that even the people who made the observation pointed out that it was just an association and there was no causation proved. So that was the situation up until about 1980. And then things changed because this gentleman, Harold Zerhausen, came along. And I'm sorry, that's a very bad copy of the actual article, but he speculated in an article in 1976 that because genital warts were very common and caused by a virus which the equivalent virus in animals caused cancer, maybe the genital wart virus caused cancer in women. It turned out that that was not true, but what he did go on to show with his colleagues, Lutz Gissmann and Andreas Kleinhardt particularly, was that there was a virus in the cervix of women with cervical cancer that looked very like the wart virus that caused genital warts. And remember, in those days, the technologies for looking at genetic information were very primitive indeed. And he managed to make an observation that he could isolate bits of the genetic information of a virus that looked like the genital wart virus in cervical cancers and published that. And if you like, that was the beginning of the end of the story about why cervical cancer might be caused by a virus. So that then led to the question, what can you do about it? So he didn't think of this, but somebody else did. He wondered if you could, this is a gentleman thinking about how you might control wart virus. And according to Facebook, he determined he would shoot his own warts off. That's not a recommended treatment. Unfortunately, on Facebook, 560 people recommend it. But it would seem more logical to actually prevent the infection in the first place. I have to stress, by the way, that when I only slightly joke about this business of shooting warts off, when we actually treat cervical cancer and cervical precancer, basically that's what we're doing. We're using destructive therapy to get rid of the problem. And obviously that's not a very sophisticated approach, although it works for the majority of patients. However, the idea of preventing it seemed like a good idea. Now, that was made possible by Harold Zerhausen's observations back in the 1980s that these viruses, papillomaviruses, were responsible for cervical cancer. What he did not know, and what we now know, is that these infections were extremely common. He thought that this was a very rare infection which commonly caused cancer. What we now know, and this is looking at data from college students, time zero is when the students came to college, and this is the first 3 years at college, and this is the accumulation of infection with the virus we now recognize as responsible for the most of cervical cancer. After 3 years, 50% of the college students had caught this virus. So that's fairly typical of all the studies that have been shown since then. They didn't know they had the infection. It was totally asymptomatic. It just was there. And the interesting thing for the majority of them is, now this is looking again over a 5-year period with 100% infected here, over 5 years, 99% of them get rid of it. So they never know they've had it, but during all of this time, of course, they're infectious and can pass the virus on. And we now know that there are 2 viruses which commonly cause cervical cancer, type 16 and 18. These are data for HPV 16, the commonest one, and that basically most of the infections get better, but some of them will progress to cancer. Importantly, what we now also recognize is that there was a pre-cancer stage, which could be seen by screening, in other words, what we see on the pap smear. And that information, of course, is now used to help prevent cervical cancer. But it also made the trials possible that were done to prove that the vaccine might work. So I'd like to introduce you to this guy. This is Jan Zhu, who was my partner in developing the technology for the vaccine that we together came up with in Cambridge. We were visiting there on sabbatical, and we started thinking about how we might study this virus. I was interested in how the body's defenses against infection might control this virus. He was interested in how the virus might cause cancer. We both knew that we couldn't grow the virus in the lab. And that normally is how you develop vaccines. You grow up the cause of the illness in the lab, render it safe in some way, and make it into a vaccine. But we couldn't do that for papillomavirus, so we decided that we would try to build the virus. And Jan had one set of expertise in that area in terms of cloning genes into expression vectors, and I was interested in the process of isolating virus particles from cell culture, and that enabled us to develop a vaccine. So that basically he and I built these things which look like these things. This is the infectious virus. This is the non-infectious variant of the virus where there's nothing inside it except the shell. And basically it's just one protein from the virus which self-assembles to make these virus-like particles. The really interesting thing was that that happened spontaneously when we got the recipe right. Why is that important? Well, if we'd had to actually stick all the bits together, if you like, the 360 copies of the L1 protein to make these virus-like particles using any energy-dependent process, it would have made the vaccine impossible to make commercially. This bit of work here, between here and there, was done with the help of a very generous philanthropic donation from a farmer up in Cairns in the Atherton Tablelands, who gave us money for equipment which enabled us to make these virus-like particles. What we actually did was take stuff from a clinical lesion. This is a photograph of the neck of the womb here, and this white wrinkly bit is a bit that's infected with papillomavirus. If you'd scraped the cells off that, they would have looked like the abnormal ones I showed you earlier. And basically, we took the virus from there, cloned out the gene which encodes the coat protein, the outside of the virus, if you like, in a system which enabled it to self-assemble. And that was the trick that other people had not tried or had tried and failed. So we used a particular sort of cell. It was actually a monkey kidney cell when we did it, first of all, but it was an important point was that it was a mammalian cell, and we got these virus-like particles, which was very nice. It took about a year, I must stress. It didn't happen overnight, but we eventually managed to get it to work. And then out of that, we could produce a very conventional vaccine. This is the stuff to make the immune system wake up. This is the stuff that it needs to see in order to make a defensive antibody against the virus. And it then became the vaccine that we now know as Gardasil and Cervarix. And I had the privilege of giving the first dose of that vaccine in Australia, the first official dose, to the daughter of one of my colleagues in the lab. She volunteered to have it done in front of the cameras so that we could have a story for the media. That's important too because it helps with fundraising, there's no doubt. Another thing that helps with fundraising is if you can develop a patent position on the technology, and we did do that. Now, people start talking about getting in bed with industry and commercialization being a bad thing. Commercialization is an essential thing if you wish to get a product out there. If you can't find somebody to make it for you, and remember, we were making teaspoonfuls and we needed swimming pools full. If you can't get somebody to make it for you, and if they can't make money out of making it, then there will be no new products. So we took a patent position out for the University of Queensland using the bits that were unique to the process, the things that other people would not have automatically thought of. And fortunately, that patent was eventually granted in our favour. But the most important point was that there was a patent, because without that patent, then the companies that make the vaccine would not have got interested. And that virus-like particle technology was, when we came up with it back in 1990, nobody else was using it. Since then, as you can see, the technology, not the vaccine for cervical cancer, but the technology as a technology for making new vaccines, has become rather popular, with over 200 citations in the Index of Medical Research Citations, Medline, in the year 2015. And that's still increasing. Every new vaccine that people are making now uses a virus-like particle technology. So that process from here to there took 15 years, during which time Merck and GSK came up with a technology for scaling up the production of virus-like particles, doing the clinical trials, and getting the vaccine, if you like, proven safe and effective. The reality is that this step here cost about $150,000. That step cost $2 billion. And that's why you need a commercial company, because they have to take that risk. Of course, we started the story off and published it as soon as we had done the work, because that's what scientists have to do to make a living. We filed the patent first, but we got the publication very shortly thereafter. And then everybody started copying us. So that these guys in America copied us the following year. We taught them how to do it. And these guys did it. And this guy, they're all friends of mine, by the way, Bob Garcia, and John Schiller and Doug Louie, who did this one, had to withdraw it because it didn't work, and went back and proved they could do it the way we taught them how to do it. And then this guy copied us as well, Massimo Tomasino. He's a good friend of mine. Most importantly, he did it in the system that we now actually use to make the vaccine commercially, in yeast, because that's a very effective one for doing it. So I now have to make a disclosure of conflict of interest. I'm going to talk about something that I have a patent interest in, and of course so does the University of Queensland. So to distract you from that pleasant thought, I'll put up that picture of me, which was on the front page of the Weekend Australian in 2006. I came down to breakfast one morning in a hotel in Sydney and couldn't understand why everybody was looking at me rather closely. And then I have to point out that I think that was probably the vaccine they were talking about there. And I've had to point out to my children that that's not a title that they're going to inherit from me. On the other hand, this was what Cosmopolitan magazine thought about me at the same time. And I really hope it was the vaccine they were talking about there. At any rate, the trials of the vaccine involve very large numbers of people, because the vaccine has to be given to people before they get the infection. And if only 1 in 100 out of those people are going to go on to get a pre-cancer or cancer, then only one person in every 100 that you recruit to the trial will give you useful information. And this is another sort of philanthropy that's really important to understand. It's the philanthropy of taking part in clinical trials. We have to encourage people to take part in clinical trials, because we cannot advance medical science without that. And very large numbers of women took part in these clinical trials entirely voluntarily. And what basically the trials showed was that prevention of pre-cancer, these things labeled CIN here, was affected in about 100% of the women that got the vaccine. So that this was, of course, what we wanted to see. The message that was important was that it only worked in the women who did not have the virus when they came into the studies. And the women came into the study with the virus, and the natural history of the infection did not change in the woman that came in already infected with the virus. And of course, the vaccine only works in those people to protect them against the type of virus that the vaccine protects against, which is self-evident. Type 16 and 18 is what's in the vaccine. The important thing to realize is that that vaccine only protects against 70% of cervical cancer. But the vaccine that we now have in Australia, Gardasil 9, includes 9 virus types and protects against about 98% of cervical cancer. So that's a big step forward. It uses the same technology, but it covers more virus types. Of course, there was quite a lot of protest at the time about a new vaccine for sexually transmitted infection, because let's face it, this is a sexually transmitted infection. Our then health minister went public and said that he would not give the vaccine to his daughter because he thought that it would increase the chance that they might catch a sexually transmitted disease, which seemed a bit sort of counterintuitive. But he changed his mind eventually, thank goodness, and the vaccine was, of course, licensed in Australia first in 2007, and we were the first to introduce public health programs where every child was offered the vaccine, first of all, every girl, and then from 2012, every boy as well. And that has been the benchmark standard now around the world, is to try and achieve that. So thinking about actually getting a vaccine out there, the story that I told you at the beginning, first of all, you have to define the problem and recognize that the virus caused cancer, develop the science through recombinant VLP technology, find commercial partners, scale up the manufacture, demonstrate it's safe. And then going back to that first slide I showed you with the several stages in the triangle, this is the most important one, educating the profession, the public, government about how the vaccine should be used. And that's an ongoing process even today. What I'll tell you a little bit about is what's happened since then, since 2006. So confirming field efficacy is really important. I've already told you we have a better vaccine, and then I'm going to talk a bit about global deployment of the vaccine because that's really important. So these are data from Australia, just looking at what happened after the vaccine was introduced in 2007. And we're using genital warts as a benchmark for what the vaccine is doing, because genital warts are also protected against by the vaccines, at least by Gardasil. And we know that genital warts appear very quickly after you get the virus infection. They're there within 6 months. And what happened after 2007 is that in the group of people of the right age for vaccination, the under 21-year-olds who got vaccinated, there was a dramatic reduction in genital warts, such that people under the age of 25 simply don't present with genital warts in Australia anymore. It's a vanished disease. If they have got them, they haven't been vaccinated and they've gone overseas somewhere and got them there. But on the over 30s who didn't get vaccinated, no change in the incidence of genital warts. Now this was in women. Oops. This is in men. And of course the men were not vaccinated in 2007. They didn't start immunizing boys till 2012. But the same reduction in genital warts occurred in the boys. The reason, of course, is that of this great phenomenon of herd immunity. If somebody else is protected by vaccination, they protect you even if you're not protected. And of course, with 70% of girls immunized from 2007, even although no boys were immunized, there was a dramatic drop-off in the incidence of genital warts in boys. Now, one important point to make is that these vaccines are not therapeutic. If you've already got the virus, they don't do anything. So this is looking at the protection level that the vaccines have given in Australia, across the Australian community, according to the age that the girls were immunised at. So that the girls that were immunised at the age of 14 mostly had not been exposed to the virus. And the level of protection was about 75%, which is about, remember that HPV 16 and 18 together cause about 70% of cervical cancer. So that's what we would expect, 70% of protection against abnormal pap smears. But for those who were 17 when they were immunised, the protection is much less. And this simply reflects the fact that most of these people have already been exposed to and got the infection and therefore can't be protected. And so the protection is significantly reduced. We do need something for those that are already infected, but I will spare you that topic today. That's what I really am interested in now, and I will get rather passionate about it, and you'll still be here at 3 o'clock in the morning. So I'll stick with just thinking about improving the product. The 9-valent vaccine, of course, is now available, and if you look here at the protection, comparing the vaccine that we had with the 4 types in it, because that was Gardasil and then Gardasil 9, in the study, this was a head-to-head comparison. 30 women who got the 4-valent vaccine out of 6,000 got a significant pre-cancer, and only 1 who got the 9-valent vaccine. So the efficiency is very high at protecting against a whole range of extra types that can cause pre-cancer. But the most important thing I want to talk about is enabling global use, because this is where we really need some philanthropic help and also some serious thought. This is a really disappointing picture. The countries in green on the map are all the countries where there are programs to protect against cervical cancer through vaccination. The blobs in purple are where all the deaths from cervical cancer occur. And you can see there isn't much of a match-up. So the question is, why are we not getting this vaccine into the developing world? Well, the answer is we have to find out. So we go out and we decided we would do a study in Vanuatu to look and see if we gave the Vanuatu government vaccine free of charge, could they effectively deliver a program? And Vanuatu is a bit of a challenge in that regard. It's a country which has got the highest gross happiness index in the world. It also has got the second lowest gross domestic product in the world. It's a broke country and everybody's very happy. There's a message there. So give generously to medical research and you'll feel much happier. It's 50 islands, 250,000 people, 25 doctors, 91 parliamentarians. Bad ratio, that. Do not encourage that ratio. Canberra would be solid parliamentarians. It is, isn't it? One vaccine fridge running off a very unreliable electricity supply. So this is a typical challenge for developing world countries to get a vaccine program out there. Most importantly, we surveyed 500 well women walking the streets in Port Vila, the capital city of Fatty Island in Vanuatu. 5 of them had cervical cancer and didn't know it. Another 17 had the pre-cancer and were going to go on and get cancer and didn't know it. So that gives it about the highest instance in the world of cervical cancer and pre-cancer amongst any female population. So we embarked on an education program, having given the country the vaccine, so to speak. And the education program was run by the Vanuatu people. Here's the district nurse. And here are the teachers from the school, the pupils from the school, the parents of the children, all there being given an explanation what this was all about. We wanted to test a hypothesis that it would be a good idea to give the kids a reminder so they would come back and get a second and third vaccine. The study failed because they all came back regardless of whether they got the reminder or not. We got a very high return rate, almost as high as in Australia for covering the island, but higher in terms of getting the second and third dose than we were managing even in the opt-ins programs in schools. And that we cluster randomized by village, and there was no difference as to whether they got a reminder or not. They all came back and got the vaccine. The message is that the women there understand cervical cancer. They know it's a lethal disease, and they know they want to do something to protect their daughters. So there's no problem getting a vaccine in place if you make sure the vaccine is available. The country can deliver it, and the people actually want it. Similarly, in Bhutan being a nice small country next to Nepal, again, a young population, the second happiest country in the world, the third lowest GDP country in the world, and cervical cancer, the commonest cancer in the population there. They decided they wanted a vaccine program. In fact, this woman here decided she wanted a vaccine program. She is the royal grandmother, and she's now 87 years old. Charming lady. She decided, having heard that India was going to try a vaccine program, that Nepal should get it. So she told this gentleman and this gentleman, the education minister and the health minister, that they were to deliver a program in the country. First year they did it, 100% coverage of girls of the age of 12 to 14, or so they claim, but I think it's probably true. They then made a bad mistake, and we learn from mistakes. So the first year they gave the program through the schools. The second and third and fourth year, they gave it through health centres. Health centres mean the kids had to walk between 3 and 4 hours to get to a health centre to get their vaccine. And needless to say, the pick-up rate for 3 doses of vaccine dropped quite dramatically. So they went back to a schools-based programme in 2014, and the coverage went back up again. So it can be done. It can be done in Vanuatu, it can be done in Bhutan, so it can be done. Critical thing is to make the vaccine available, preferably free of charge. So how are we doing with cervical cancer control globally at the moment? Well, let's think a little bit about the problem. First of all, we've delivered 200 million doses of the vaccine globally as of last year. It's probably about 240 million now, 10 years since the vaccine became available. At the moment, there are 25 million women, mostly alive, who will die of an HPV-induced cancer globally. That's the challenge. These are the people who are already alive and who in their lifetime will develop cervical or esophageal or whatever cancer caused by the virus. The theoretical benefit from the vaccine delivered so far, the 200 million doses, would be 2 million lives saved. 1 in 100 people who get the virus go on to get the cancer, and almost everybody is exposed to the virus. So that's what we should see as the benefit. But the reality is that's what the benefit is. And the reason for the fact that we've only really saved 10,000 people is because the vast majority of vaccine has been delivered in the developed world, where there are perfectly good screening programs which women take part in, which would prevent cervical cancer anyway. And only in the developing world are we really making much of an impact on the disease, and most of the vaccine is not going there. So we need to think about how we could sort the problem out. Now, one problem, obviously, is the cost of the vaccine. But another problem is that we have to deliver the vaccine 3 times. At least that was the policy originally. And the clinical trials were done with 3 doses of vaccine. But one really interesting thing emerged from those people who took part in the original clinical trials but actually only got 1 dose of vaccine. And they were the ones who didn't come back for the 2nd and 3rd dose, but were nevertheless followed up on. And when you actually look at the efficacy of the vaccine, there isn't any difference between the people who got 3 doses and the people who got 1 dose in terms of prevention. Admittedly, the numbers are small, only 292 versus 11,000 in that group, but the protection efficacy against infection at 5 years is very high. So you could well argue that if you treated this as an epidemic disease and went out and did programs of immunization rather than developing basically an immunization campaign rather than a regular vaccination program, and just immunized everybody, you could break the cycle of transmission and get rid of the infection altogether on a global basis with 1 dose per person, which would, of course, reduce the cause considerably. So that really is what I would put forward as one part of the solution to the problem. So that these are the current issues. We have to decide what age to immunize, and that is country-specific. In Vanuatu, it's 10 years. In Australia, it's 12 years. In the Middle East, several countries, it's 14 years, because it depends on the age at which sexual activity starts. Where to immunize? Well, I've shown you that schools are the best place. That's where we do it in Australia and where we'll continue to do it worldwide. I would like to argue we just should give 1 dose, and probably HPV 16 and 18 would be enough to make a real impact on the disease if we have to use a cheaper vaccine rather than the more expensive one. And we could probably get off with only immunizing girls, because the data from Australia suggests quite strongly that the herd immunity, if you immunize 70% of girls, is sufficient for protection. So basically, I would be thinking that we should be campaigning for single-dose mass immunization programs on a global basis at the moment. Can we pick out the people that are at risk, the 1% that are going to go on and get cancer in advance? Well, the short answer is not really. We know that there's no particular connection with the things that people do. It's not whether they smoke. It's not whether there's different variants of the virus. It's not oral contraceptive use. It's strongly linked with genes which determine your immune response. And this is work that was done here in Brisbane and elsewhere by Matt Brown and myself and a number of our colleagues worldwide. And we found basically that we could map this as a genetic risk. But it's cheaper to vaccinate than to find out if you have the genetic risk, and it's much easier to do. So the best bet is to just immunize and not worry too much about whether the people who have got the disease. It's true that the people in the highest 10% of genetic risk have got a very high chance that they'll get cervical cancer. The people in the top 20th, the highest 5%, have a 1 in 5 chance they'll get cervical cancer in their lifetime. But it's still not enough to justify screening for risk. So what we need to do is to make a case for worldwide and universal vaccination, and this will be my last point for today. So I'm going to compare polio with cervical cancer. because we have a vaccination program worldwide to prevent polio where we're trying to eradicate the disease. And I'll remind you that both poliovirus and the papillomavirus only infect humans, so that the principle is the same for both. Lifetime risk of getting the infection with the virus pre-vaccine was about 50% for polio, about 50% for HPV. Lifetime risk of serious disease, 1% for paralytic polio, 1% of the people that get the polio virus will get paralytic polio, 2% of the women who get papillomavirus will end up with surgery to prevent cervical cancer. Lifetime risk of death, about 0.1% of people, 0.8% for cervical cancer, 8 times higher. In the last big epidemic of polio in the United States, when the new vaccine, the polio vaccines became available in 53, 54, 3,100 people died and 21,000 were left with paralysis in that 1 year. In 2005, which for the USA was the last year before vaccine became available and was a very average year for cervical cancer, 3,900 women died of cervical cancer and 12,000 were diagnosed with cervical cancer and needed treatment for it. And remember, this isn't a country with a screening program which is available for all women to help prevent them getting cervical cancer. So this is basically showing that the screening program is insufficient. So you can see, at least in my opinion, that there is no doubt that cervical cancer is as big a public health problem as polio ever was. And of course, we now have effective vaccines against both of these viruses, and the safety of the cervical cancer preventative vaccine, the papillomavirus vaccine, is at least a log order of a magnitude better than that of the poliovirus vaccine, which in about 1 in 100,000 people causes vaccine-induced paralytic polio. So I would argue very strongly that based on this evidence, we should be advocating as strongly for a vaccine program worldwide to prevent cervical cancer as we're currently doing to prevent polio. And we hope to eradicate polio within the next 10 years, but we could eradicate cervical cancer in this decade. So it only remains to acknowledge that this is work that's been done with funding from a very wide variety of people, and also the work in the lab, although I haven't talked too much about my lab work today, have been done by a very large number of people as well. And when they're doing the lab work and these guys are funding it, I sometimes manage to get off doing some skiing. And those are my tracks. So thank you very much for your attention.
Thanks so much, Ian, for a wonderful story and a fantastic lecture. Look, we actually have time for some questions for people who have got burning questions for Ian. And yeah, Jeff.
So Ian, what do we do with the screening program? How do we manage the interface between the vaccination program and the screening program? It's one of the things that concerned us as we looked at Gardasil for funding.
Well, of course, ideally, well, first of all, we've had to change the screening program because of the vaccine program. Imagine yourself as a screener sitting there looking at slides taken from the cervix of women. Pre-vaccine, 1 in every 40 of those would have abnormal cells in them. You would look at about 40 a week, so you'd go to work each week knowing that one of the things you looked at during that week would be abnormal. With post-vaccine, that figure will drop from 1 in 40 to 1 in 1,000. So you know that you'll be working for half a year before you find an abnormal smear that you've got to do something about. The job would become unsustainable. And more importantly, the quality assurance becomes unsustainable too, because we rely on a certain percentage of smears being read as positive to indicate that a lab is actually doing its job right and doing the screening. So what we're now going to do is screen for the virus. And we're going to start screening at an older age group, because we know that the cancers don't come till later, although the virus may be there earlier. And we want to wait until most people have cleared the virus away. So by the age of 25, most people have had the virus, got rid of it, and we're only picking up the people that have got persisting infection. And then if the virus is there, we'll watch them more carefully and do the test for looking for the cancer. But there's a bigger question there, is should we put more effort into the screening program or more effort into the vaccine? And on a global basis, screening is really difficult, but vaccines are really easy. So that globally, the solution is vaccinate and don't worry about screening.
Okay.
And if you catalog
Yes, I could tell you that it's a failure so far, but that's a little unkind to the people that are doing the work in the lab. Look, it's obviously desirable to have some way of getting rid of the infection once you've got it. The challenge is to get something that can do that that is cheap enough and simple enough to use on a global basis, because that's the real problem. And in this country we can screen, but in sub-Saharan Africa and India, we're never going to be able to screen, I don't think. The logistics will defeat us. So we are working on an immunotherapy. The first trial of immunotherapy we did was back in 1998. It didn't work. Subsequently, we have spent a long time working in animal models. I keep telling my students that mice lie all the time, by the way. They don't tell you the truth. But we use the animal models because it's easier at some level than trying to do things with human samples. And we've learned why it's really difficult to get rid of this virus with immunotherapy. And we've got some fairly clear answers to that, and sufficiently clear answers now that we are planning to go ahead well, we're actually doing a trial at the moment, a safety trial of an immunotherapy, which we hope will work. And if the safety trial, which will be finished by about the middle of next year, is satisfactory, we'll go on to a real trial of a combination of immunotherapies, which we believe from the data we have from the animals will actually work in humans. So that will it'll be an early phase clinical trial in, not funnily enough, cervical cancer, but rather in oropharyngeal cancer, which is another cancer caused by papillomavirus, for which we need better treatments.
If there's no other questions, join me in thanking Ian for a great talk, and John for